Hello, everybody. Chapter 4 focuses on sex determination and sex-linked characteristics. We will discuss these two features in two different recordings. This first one will focus on sex determination. Previous chapter discussed Mendel's principle of segregation and independent assortment, two principles that can explain nature of inheritance of many different traits. However, there are many traits which are inherited by much more complex mechanism, and therefore there are many exceptions and extensions from Mendel's principle of inheritance. One of the major extension is inheritance of sex-linked characteristics. So what are sex-linked characteristics? Those are characteristics or traits which are determined by genes located on sex chromosomes. To understand inheritance of such traits, you have to first understand how sex is determined, and that's what we will discuss now, because there are a number of different mechanisms in the nature how sex can be determined. So sex can be determined at several levels. It can be determined chromosomally, genetically, or environmentally. So let's first talk about chromosomal sex determining system, which is outlined in these first three lines. So what does it mean when sex is determined chromosomally? It means that pair of chromosome, which we call sex chromosome, differ between males and females. Do you recall that autosomes come in pairs? Homologous chromosomes are found in one pair, and basically these homologous chromosomes have the same size, the same shape, and host or carry the same genes. This does not apply to sex chromosomes. So let's start with a simple system, which is XXXO system, which was also described as a first. You can find the system in grasshoppers and some insects. In XXXO system, females are homogametics and males are heterogametics. However, heterogametism here means that males possess only one X chromosome. So during gamete formation, half of gamete formed by males will carry X chromosome and half will only carry autosomes and no X chromosome. Second system which you are familiar with is XXXY. You are familiar with it, of course, because it's how sex is determined in humans. All mammals carry XXXY chromosomes. This system you can find also in amphibians, reptiles, fish, and some insects. In XXXY system, females are homogametics and males are also heterogametic as in previous system. However, here males carry two chromosomes, X chromosome and Y chromosome, and it is Y chromosome which determines maleness. Now, I would like to bring your attention to one fact, and it is, even so, we classify XXXY as a chromosomally determined system, it is at the same time also genic sex determination. Why it is? Because also the two chromosomes are phenotypically different. They also carry different gene, and it is a gene, SRY gene, which is localized on Y chromosome, which determines maleness. So it is combination of chromosomally and genic sex determination, chromosomal sex determination and genic sex determination. And we will talk about this system a little bit more later on. Let's now go to the ZZ-ZW system. In this system, females are heterogametic and males are homogametic. You can find the system in birds, in amphibians, and some reptiles. keep in mind that when we will talk about sex-linked characteristics, whatever applies for XXXY system will be opposite, the determination will be opposite for ZZZW system. In addition to these three chromosomally determined sex mechanisms, we have also additional one which is called haplodiploid. In haplodiploid system, males are haploid and females are diploid. You can find this kind of sex determining system in bees, wasps, or ants. What does it imply? How does this reflect in gamete formation? Female is diploid, so females are generated by fertilized eggs, while males are produced from unfertilized eggs. So when female form gametes, it has to employ meiosis, while males, since they are already haploid, they produce gametes by mitosis. And this has implication on inheritance, which we will discuss a little later. So now let's go back and let's discuss genic sex determination. In this system, you cannot see in two individuals, in males and female, obvious difference in chromosomes. So under such system, sex is completely determined by genes located on the chromosomes. You can find this system in plants, some protozoan, and some fishes. Now, keep in mind that even in chromosomally determined sex systems such as XXXY, it is actually genic sex determination because it is a gene or genes that are localized on that particular sex chromosome which determine maleness, Y chromosome, which are involved or responsible for sex determination. The last system which I would like to point out very briefly is environmental sex determination. Under this sex determination, the sex of the offspring is influenced by environment. An example you can find in turtles where warm conditions, warm temperature produces more females, while cold produces more males. And you find similar system in alligators, just in opposite ways. In alligators, it is a warm temperature which produces more males. So these are the very basic system by which sex is determined, and we will actually talk about another system which is very particular, which you find in fruit flies. So let's now talk about sex determination in humans. In humans, Y chromosome is one which determines sex. In other words, if individual does not have Y chromosome, if it possesses X chromosome, XO or XX, it becomes female. However, if individual possesses Y chromosome, it will develop in male. Of course, there are certain exceptions, very rare disorders, which we will talk about later. However, under normal circumstances, XY develops in male because Y chromosome carries SRY gene, which is gene that determines maleness. SRY gene actually is turned on very early during embryogenesis, and it does determines a development of testes and results in production of testosterone, which drives male development. Let's now go back and look on X and Y chromosome here in this picture. First things which should occur to you is that these two chromosomes are different in size, different in shape. So how they can align during metaphase 1? And the answer lies in so-called pseudo-autosomal regions, which you can find at the end of both X chromosomes and Y chromosome. The pseudo-autosomal regions are homologous regions which allows X and Y align together in metaphase plate. Not only that, they also allows very limited recombination between X and Y chromosome. Now, I will step back and talk a little bit about importance of Y chromosome. Why Y chromosome, or how we actually know that it is Y chromosome that determines maleness. In fruit flies, Drosophila melanogaster, sex is determined by genic balance system. What does it mean? Even though fruit flies have Y chromosome does not carry genes which would determine maleness. The sex is determined by balance of female determining genes which are located on X chromosomes and male determining genes which are located or localized on autosomes. So the sex is determined by a ratio of X chromosome to set of autosomes. Let's say fruit flies, normal diploid fruit flies has 8 chromosomes. Out of this, you have 2 sex chromosomes and you have 6 which are autosomes, which comes in 2 pairs. So this is 2n. Now, if you have fruit flies which is XX and is normal diploid, how do you calculate sex? You divide number of X chromosomes, which in this case is 2, by number of sets of autosomes, which is, this is diploid, so there are 2 sets, so you divide it by 2 and ratio is 1. If a ratio between X chromosomes and sets of autosomes is 1, fruit flies is female. If this ratio, if you have fruit fly which is XY and is diploid, so it has 2 sets of autosomes, so the ratio will be, you have only 1 X chromosome, so it's 1/2, which is 0.5. Under such circumstances, the individual is male. Keep in mind that sometimes you can find fruit flies which are triploid, or sometimes there is even different ploidity which you can find in fruit flies. So if you calculate ratio between X chromosome and sets of autosome which falls between 0.5 and 1, those fruit flies will be intersex. If the ratio is higher than 1, you are looking at meta-female. If the ratio is below 0.5, you have meta-male. So let's look at some abnormalities in chromosomes number or chromosomal number which results from non-disjunction during meiosis. These disorders will clearly illustrate importance of the Y chromosome in human sex determination. Let's first look at XO constitution, which means that that individual has only 1 X chromosome. This occurs in 1 out of 3,000 female births, and it is known as Turner syndrome. So XO will develop in female. These females possess only 1 X chromosome, and usually they are shorter, they have low hairlines and broad chest, but they do not have normally any problem with intelligence. Their intelligence is normal. However, most of them are sterile. Keep in mind now, can we have OO individuals or OY? No, we cannot. X chromosome carries genes important for embryonal development, so embryo which do not carry any X chromosome would abort, would not develop. However, let's step back and talk about importance of Y chromosome, and you can see it in Klinefelter syndrome. In Klinefelter syndrome is a result of non-disjunction of X chromosome, and you can have individual which is XXY. If you look XXY in fruit flies, it would be female. However, in humans, since Y chromosome determines maleness, this individual will be male. Also, Klinefelter male are usually they have small testes and reduced facial and pubic hair, so secondary sex features are not developed properly, and they are usually sterile. They have normal intelligence and they function normally as a male otherwise. A little bit slightly different are further non-disjunction when there are 3 or even more X chromosome. If there is this overdosage of X chromosomes, basically it reflects in the intelligence. Now, another possibility which you can have as a result of non-disjunction are polyfemale. Poly-X female overdosage of X chromosome actually result in intelligence problems and certain retardations. So, if you look in humans on these disorders resulting from non-disjunction, meaning problem in segregation of chromosome in meiosis, you can see that whenever you have Y chromosome, these individuals will develop into male. The last thing I would like to discuss are certain disorders in development of sex due to the mutation in SRY gene or due to the mutation in gene which influence development of sexual characteristics. First thing which I would like to point at is essential role of SRY in maleness in the development into male. The direct proof for such role we can find in Swyer syndrome. In this syndrome, XY individuals which normally develop into male will have female characteristics. So what is the reason? In these individuals, SRY gene has been lost, let's say either due to deletion or due to mutation which render this gene completely non-functional. So this is a direct proof on the importance of SRY in development into functional male. Another very rare syndrome might occur when SRY gene is translocated on autosome. In such case, the individual who inherits this autosome carrying SRY gene will develop into male if translocated SRY gene is functional, even though that person might contain XX chromosomes. So these two are examples which show directly importance of SRY gene in the development of males. However, you have to keep in mind that although SRY is a primary determinant of maleness, there are many other genes which might be located on X chromosomes or autosomes which influence development of male characteristics. Let's talk, for example, about one example, which is androgen insensitivity syndrome. This syndrome is caused by mutation in androgen receptor. Do you recall that SRY gene triggered development of testes and production of testosterone? However, in order for testosterone being functional, it has to bind and activate androgen receptor. So in people with androgen insensitivity syndrome, where androgen receptor is mutated, although the level of testosterone are normal, these people develop female characteristics. I apologize, this is a mistake. It is localized. Gene for androgen receptor is localized on X chromosome. So these are just few examples of disorder which you can detect in humans, which really witness or show importance of SRY gene and genes which are related to the function of SRY in the development of male characteristics in humans.